Hi, everyone. I've changed this talk about 10 times since I arrived at the meeting because this is my first time at a CMT meeting. I'm really inspired by everything that I'm learning. And thank you for giving me the chance to give a talk. Before I give any talk, I always want to thank the members of my lab who make all the magic happen. The lab is a lot smaller now. You can Google my name in CNN if you want to read more about that. I also like to point out that my lab comes from all over the world. And I think that one thing that makes America great is that people come from all over the world here to work together to make the world a better place. And I think that's really cool. And I intentionally put Iraq and Iran next to each other and China and Japan next to each other. So it doesn't matter what your geopolitics are or what your history is. And then I also have to point out that Florida is best understood as a separate country. My lab works on a lot of different things. I was born with severe to profound hearing loss, so I have a deep passion for children with disabilities. And my lab works on hearing loss, and one of the big discoveries still not published is that psychedelics induce new synapses in the cochlea, and I'm going to talk about that a little bit later. We also study organelle dynamics in patient-derived models of neurodegeneration. And then our real jam is imaging and deep learning. So all of these projects generate tons and tons of beautiful data that we can then train AI models to help us interpret and work with. And I'm also director of the Goodell Family Technology Sandbox, which is a one-of-a-kind facility in partnership with Thermo Fisher and Nikon, where we have cryo-EM, cryo-ET, flow cytometry, mass spec, and light microscopy all under one roof with the vision of building like a biology chat GPT with these multimodal data sets. And so as you can see, I work on a lot of different things, and this is my constant state of being, and I have to justify it every day. So one of my favorite things is mitochondria. I've been studying it as a cell biologist for a long time now, and everyone knows we've heard about mitochondria in earlier talks today. And one of the really cool things about them is that they're so insanely dynamic and constantly fusing and dividing as they move throughout the cell. And you can only capture that with live cell imaging. So we develop a lot of live cell imaging tools in my lab. And, you know, not only is it very cool, it's also very important. And one of the things I noticed very early on is that a lot of the really key proteins that I was finding were regulating these dynamics are mutated in Charcot-Marie-Tooth disease. And that's how I ended up here today. And so through a series of papers, my lab has shown that mitochondrial dynamics are regulated by ER mitochondria contacts and actin filaments at those contacts. And then we also found that mitochondrial fusion is regulated by ER mitochondria contacts. And we found that this protein called mitofusin 2 that some of you have heard of localizes not just to fusion sites, but also to fission sites. And this other protein, DRP1, that typically mediates fission also localizes to fusion sites. And we also found that if you perturb any of those particular components, then one thing that happens is mitochondrial DNA is released from the mitochondria into the cytosol, and that activates the C-gas sting innate immune pathway. And I'll talk a little bit about that later. But in any case, we now have this model where we have these key mitochondrial dynamics proteins, mitofusin 2, INF2, and RAB7, all of which are Charcot-Marie-Tooth genes converging at ER lysosome mitochondria actin contacts. And if you perturb them, you end up with mitochondrial DNA released into the cytosol, activating the antiviral response. So I became very interested in mitofusin 2. We collected many patient cell lines with different mutations in mitofusin 2 that have a big spread of phenotypes. They all have, some of them have really fragmented mitochondria, others have really fused mitochondria. But what we saw is that they all have reduced motility, and I'll show you some of that. And then through this work, someone heard about me, and I'll talk about that in a moment, but our model is that all of these mutations impact the movement of mitochondria. And the reason it causes specifically a peripheral neuropathy and not just systemic disease is because those peripheral neurons are so long. And we actually did some calculations, and we found that in a normal, healthy human, it could take weeks to months for a mitochondria to travel from the cell body all the way down to your toes. And if you have a disruption in mobility, it could be even longer or maybe not ever make it. So we think that's a key pathology is this motility defect. And through my work, I got in contact with a family. It was one of the most emotional meetings of my life. I'm just a scientist. I'm not trained to talk to patients. And they had heard about our imaging work and wanted us to screen drugs on their daughter's cells. And I kept it together during the call. And then as soon as we hung up, I started crying. And Terry really triggered that in me again. So I'm very passionate about this, and we're going to do everything we can. We're basically throwing the kitchen sink at this problem in my lab. And this is me with her father, and this is her, Michal. And one of the first things that happened was I reached out to UCSD's sequencing team and asked them, can they help us sequence her cells so that we can start designing gene therapies? And they said, yes, in fact, my daughter has Charcot-Marie-Tooth disease. And it was the admin, and she joined my first Zoom call with the team. At the end, I asked her, so what is the mutation your daughter has? Turns out she has the exact same mutation as Michal, which is a really rare T206I mutation that has been seen like 8 times in the literature. And we have since then made a rat model that has the same mutation. But the first thing we do is we work in vitro with patient cell lines. So we have developed this spheroid model system where we can take patient cells, turn them into iPSCs, and then turn those into motor or sensory neurons. And that's what the motor neuron culture looks like. And immediately we could see that the CMT2A patients, the axons did not grow out as far. And we can grow them in cover slips or in microfluidic patterns so that they simulate a nerve like what you might see in vivo. And then once we have these cultures going, we can then image mitochondria and lysosome trafficking up and down. We know exactly what direction they're moving in. We know exactly how fast they're moving, and we can characterize the mobility in disease and in health. And what you can see is that in the CMT2A patients, the mitochondria are not moving as well. And one of the big surprises that we came across was that also lysosomes are not moving. In fact, lysosomes are more impacted than mitochondria. Not to emphasize that everything I'm showing you is not published yet. So that means that we want to look at all the organelles, not just because what else might not be moving. So we're developing this label-free approach where we can see all of the organelles in the cell and capture fluorescence plus label-free imaging so that we can completely phenotype these cells as we treat them with different drugs or gene therapies. And even without fluorescence, we can immediately see a phenotype, or what we call a fingerprint, that we're now using to collect from a whole constellation of Charcot-Marie-Tooth patients so that we can train AIs to identify the fingerprint and then reverse it with different therapies. And one of the, this is kind of compressed, I'm sorry about that. We did find that all of our patients have elevated levels of cytosolic mitochondrial DNA to bring us back to this point that if you disrupt organelle dynamics and interorganelle contact, you're going to end up with mitochondrial DNA getting released. So again, through this work, I ended up getting in touch with Sridhar Natarajan. Some of you know him. He was head of science at Sanofi, and he has become really interested in some of Gerald Dorn's pioneering work with mitofusin activators, which is a very logical target for mitofusin mutations. And so we decided to test these mitofusin and piperine derivatives in our patient cells. And what we could see is that they restore not just mitochondrial movement, but also lysosome movement. So these compounds are really interesting. And most importantly, they also restore mitochondria in the distal end. So what you're looking at here on the top and left is mitochondria in the proximal region close to the cell body. And then on the bottom, you can see them in the distal region. And what you can see is that in the patient, there's far fewer mitochondria. And when we treat with this piperine derivative, mitochondria in your toes basically are restored. So we're restoring transport of mitochondria and lysosomes out there. And most importantly, we can see that the axons then grow back out to a more normal length. So we're restoring organelle mobility and we're restoring axon outgrowth. And I mentioned psychedelics for synapses in the ear. We decided to see if they can also help sensory neurons from patients, and we see that in fact they do. And I think that this increased plasticity and rejuvenation of some of these compounds can be combined with gene therapy in the future to have better efficacy. So I mentioned we made a rat model. We have confirmed already they have a grip defect. They also have a auditory neuropathy phenotype, and they also have the reduced organelle mobility mirroring the patient cells that we see. We are designing allele-specific siRNA gene therapies. We started with the mutation-targeting siRNA, got a bunch of feedback from funders that this is only going to help 1 or 2 patients. So then we decided to look for population-wide SNPs, and we've identified a few and are actively developing those siRNAs as well, so that hopefully we'll just have a limited set of siRNAs that can treat anyone with CMT2A. And we've also made endogenous tag cell lines so we can actually see where the mutant protein goes. And I told you about the lysosome phenotype. As it turns out, this mutant protein goes to lysosomes, which is not normal, and I think an important part of the pathology. We've also developed mass spec approaches where we can measure the exact level of the mutant versus wild type protein in the tissue. So we're not just looking at RNA transcript levels, we're looking at protein levels in all of our different knockdown approaches. And then, of course, we're bringing all of the technology we have to bear to build these multimodal AIs so that we can design better medicines in the future. I'm sorry, these are all compressed. I don't know how that happened, but one of my dreams is to also take clinical data from human biomarkers, transcriptomics, and then the in vitro data and build AI models that are more predictive than what we have today. And with that, I'll thank again the CMTRF for bringing me out and your attention and take any questions.
Sorry, I'm all the way over here. Jacob White from Lowell University. I really loved your talk. Very emotional. I love how patient-centric it is. Very interesting about the lysosomes. And I was wondering to ask you, do you think that mitophagy is also impaired in the case of CMT2? And then the second part of my question is kind of a separate question, but you were talking about protein trafficking to the lysosome. And we actually see that in CMT1A because overexpressed PNP22 is diverted to the lysosome. So I'm wondering if, even though there's 2 separate mutations, this might be a convergent sort of downstream degenerative mechanism of CMT in general.
Did you say if it's also disrupting mitophagy? Yeah, okay, good. Yeah, I do have hearing loss, as I said, and yeah, I could talk for an hour and a half about all this. I tried to compress it all into 10 minutes. I skipped a lot. So there is data that mitofusin 2, which causes CMT2A, interacts with RAB7, which is CMT2B. Coincidence? I don't know, but that interaction is important for mitophagy. So our hypothesis is that these mutations disrupt normal mitophagy, and these activators can restore mitophagy. I didn't have time to show that data. And then you had another question about lysosomes and aggregates. That's a common theme in a lot of neurodegenerative diseases. I skipped over it. This mitofusin activator didn't just help CMT2A patient neurons. It also helped neurons with mutations in INF2, which is a completely different protein. And there are a couple papers here and there. I haven't looked at them too closely. There's one paper showing that it can help ALS, another paper showing that it can even help spinal cord injury. And actually, one of the things I saw in spinal cord injury is release of mitochondrial DNA activating cGAS sting, and these piperine derivatives seem to reverse that cGAS sting signaling. And then one last point. These piperine derivatives are derived from piperine, right? Sorry. And piperine, black pepper is 40% piperine. And when I told the father of Michal about this piperine result we had, because we saw efficacy with piperine in our dish, he freaked out because he said his young daughter never gets enough black pepper on her food. Like however much he puts on, she always wants more. And we're wondering if that's like a body knowing what it needs kind of thing. We don't know, but I thought that was interesting and thought I should share.
One quick question from online. Sorry, in the corner again. How far are these treatments for the likes of CMT2A, the mitofusin or the mitofusers? How close are they to clinical trials?
I haven't seen any clinical trials for CMT2A.
Yeah.
Maybe I misunderstood your question.
Yeah, no, I have a question from online just asking.
Oh, from online.
Yeah, how close are these drugs that you're testing? How close are they to making it to clinical trials? How far are they away?
Oh, I mean, I have no experience with clinical trials. That's part of my motivation in being here is to talk to people who can help us get these things into the clinic. I'm very motivated to figure it out. We're, like I said, throwing the kitchen sink. We're designing ASOs, siRNA, knock down and replace AAVs. And all these small molecule approaches. We're going to test everything we have in our rats, and whatever looks promising, I would love to put it into clinical trials, but I have no idea how to do that. I'm just a cell biologist. I don't know what I'm doing here. Yeah. All right. Thank you.
Thank you.